The next few weeks, we'll be using gravimetric analysis to determine the percent of chloride in an unknown sample. In this short pre-lab lecture, we will first clarify what gravimetric analysis is, summarize the procedure for Experiment 2, and discuss what is needed for the worksheet due after lab. Here are the learning goals we will emphasize for Experiment 2. Take a second to read them so you understand what will be asked of you for this lab. To begin, I will mention some things about lab safety and proper technique. Gravimetric analysis is a very sensitive technique, and even the oils from your fingers can impact the experiment. That being said, make sure you wear gloves. Gloves are also necessary because of the compounds we will be using that contain silver. Silver nitrate, for example, when exposed to light, will decompose to make silver solid. If on skin, this decomposition reaction will lead to a black stain forming. It isn't dangerous and will go away within a few days, but I don't think any of you really want to stain your skin, so be sure to wear gloves. Gravimetric analysis is defined as quantitative isolation of the analyte of interest from the sample in highly pure form, or in some combined form, followed by weighing the isolated analyte. Here you can see two examples of gravimetric analysis. In the first example, gold is precipitated in the pure form from the solution using sodium bisulfite as a precipitating agent. In the second example, barium is precipitated in a combined form when sodium sulfate is used as a precipitating agent. The three major requirements for a good gravimetric analysis are that, first, the precipitating agent will react only with the analyte of interest to form a precipitate. Second, it forms one and only one product with the analyte. And third, that the analyte precipitate quantitatively from solution, or in other words, that almost all of the analyte precipitates. Here is a summary of the procedure generally used in gravimetric analysis experiments. To start, there is an aqueous solution containing the analyte. A precipitating agent, here silver nitrate, is added, which results in a precipitate forming that contains the analyte. In the next step, the precipitate is filtered, dried, and finally, based on the way to precipitate, you can determine the percent chloride you had initially in your sample. In any gravimetric analysis, you should make sure an excess amount of precipitating agent is added. But why? Whenever you have a saturated solution of an ionic compound, like sodium chloride or silver chloride, part of the ions are in the form precipitate or solid, and the other part of the ions are dissolved in the solution as free ions. For some ionic compounds, like sodium chloride, the concentration of free ions in the solution is high, and therefore has a large solubility product. On the other hand, some ionic compounds, like silver chloride, have a low solubility product. Therefore, most of the ions are in the form of precipitate. However, there are still free ions in such solutions. So excess amount of precipitating agent, here silver nitrate, is added. Based on Le Chatelier's principle, chemical equilibrium goes to the left, and so we can assume all chloride ions are in the form of silver chloride and only negligible amounts of free chloride ions remain in the solution. Therefore, when you weigh the precipitate in a gravimetric analysis, you can be sure almost all the analyte, here chloride ions, are in the form of precipitate. Now, let's talk about the Experiment 2 procedure we'll be doing in lab. Before starting Experiment 2, make sure to complete Experiment 1 if you haven't already. Experiment 2 is a 3-day lab. On day 1, you're going to clean 3 filter crucibles and put them in the oven for the next lab. When cleaning the crucibles, please make sure you do all 3 cleaning steps mentioned in your lab manual. That means first wash the crucibles with ammonium hydroxide, then with nitric acid, and finally with hydrochloric acid. Make sure to dispose ammonium hydroxide left in the flask into the base waste container before starting to wash the crucibles with acids. Here you can see the setup you need for cleaning the crucibles. You need to clean 3 crucibles. You can do all 3 at the same time if you have access to enough vacuum systems. To do so, First, open the valve and connect the tube and the rubber hose with the crucible on top to a filter flask. Make sure you have some DI water in the flask before you start cleaning the crucibles. Also, make sure you rinse the crucible with large amounts of DI water after each cleaning step. After cleaning, use a permanent marker to mark crucibles, or you can put the 3 crucibles in a large beaker and put it in the oven for the next lab. Don't forget to label the beaker. Steps 3 through 5 are for the next lab after your crucibles are dried in the oven. If you have enough time on day 1, you can start doing the next step of the experiment. From your unknown chloride sample, weigh out accurately by difference 3 portions of the dried sample of about 0.5 to 0.7 grams, each to within 0.1 milligrams. Dissolve each portion in a clean, separate, labeled beaker of 150 ml of distilled water with about 1 ml of concentrated nitric acid added. Heat the chloride solution on a hot plate and add an excess amount of silver nitrate according to the procedure. Stir constantly with the glass stirring rod. The figure to the right further explains why silver nitrate is added to the aqueous chloride solution. In solution, silver nitrate breaks apart, allowing the silver to form a solid with chloride, leading to the presence of silver and nitrate-free ions left in solution. Stop stirring and let the solution settle. Check precipitate completion by adding a few drops of silver nitrate to your beaker. Place the beaker in the assigned cabinet for the next lab. On day 3 of the lab, filter the silver chloride precipitate through a weighed crucible with suction. Wash the precipitate in the beaker with nitric acid and quantitatively transfer silver chloride precipitate to the crucible. Place the crucible containing silver chloride in the oven for the next lab. In that lab, you will weigh the crucible, and based on the weight of silver chloride precipitate, you can calculate the percent chloride in your unknown sample. On the last day of Experiment 2, you will prepare 2 solutions needed for Experiment 3. So prepare an EDTA solution and a calcium standard solution. Also, please submit a zinc unknown container. From this video and the Experiment 2 procedure, you should be able to answer these questions. Please submit them on Canvas before coming to lab. After Experiment 2, you will submit a worksheet for your TA to grade. Make sure you include all of the information on this slide. If you need any clarification, please contact your TA. In your worksheet, you may have to include a Q-test. A Q-test is used if you suspect one of your values is an outlier. This slide examples how to conduct a Q-test, but if you have any questions, please ask your TA.